But yeah, it's not going to go outside of our usual meetings, so you don't have to worry about that.
Okay.
Yeah.
All right. So I guess I should start over then. What have we been up to since August? So when we finished in August for wraps, we had a process in place for basically generating simulated cell data using the Brownian bridge model. And we do that for a set of simulations, somewhere, anywhere from 25 to 100, something like that. Then we could compute Crocker plots / matrices for those simulated cell data, which was the TDA portion of the project. And then with that, we could rework the Crocker matrix into a form that could be put into a machine learning algorithm like a support vector machine, and then we could classify based on that. And the classification objective was clustering or non-clustering based on the motion or the Crocker information from the simulated cell data. So once we had that all in place, I think back in September, we were thinking about where to go with this. And we had a couple different ideas, but we decided to go with basically continuing with the momentum that we had with the machine learning parts of the project, which was we thought of basically taking that full information, Crocker matrix information, and limiting how much the support vector machine sees of it. So basically, like specifying a starting point and an ending point that aren't necessarily the starting and ending of the full Crocker matrix. So a sort of intermediate window in between the start and end.
Connor, sorry to interrupt. Can you give me a brief description of what the Crocker matrix actually, what information that has in it?
Sure. So the Crocker matrix, and it's an I don't know what the word is, but it's every letter means something, and I think it's contour realization of, gosh, I can't remember, computed K.
Yeah, nobody really knows.
Something. I lose it at the K. And then the R is RIPS complex. But basically, maybe I should start at the TDA portion, if that would be, might make that a bit clearer. So with topological data analysis, which is shortened to TDA throughout this, you basically have point cloud data, right? And you are creating connections between those points by basically creating disks around each of the points, if that makes sense. Kind of growing disks around the points. So you do this for small values, and then you make the disk larger and larger. And so you have a value that controls this disk's radius, or radius. And so you scale that up from very small values to very large values, and depending on where you cut that off, but that's known as the scale parameter. And so the way a Crocker plot or matrix works is it has that scale parameter in the y-axis, at least in the way we're plotting it. So as you go up the Crocker plot, you're seeing a larger disk around each of the point cloud, around each of the points. But our data with the cells, they move around throughout time. So in each time frame, they're in a different spot. So you have to recalculate that each time they move around, because some might have moved closer and would have gotten connected sooner. That is, they would have gotten connected at a smaller scale parameter as maybe opposed to before. And any number, and some connections might have been broken. Any number of things could have changed between the two time frames. And so we recalculate that each time frame, which in the current project right now is usually around 100 time frames. So you do that each time, and what you end up having is a matrix which has time frames and then the scale parameter again. And then you can plot that using a contour plot. And I can actually scroll down here a bit, and this is the result of that. I don't know if it has a color bar. Yeah, okay. I couldn't see it because of the zoom thing. But so basically what you're seeing here, and these red lines were added for some context for the window approach. But in the general approach to the Crocker plot, these colors are how many connections are being made, or how many connected components there are in this case. And I think this is actually Betty 0, so I mislabeled this here. So my apologies for that. But so what you can see here is the number of connected components at a very small scale parameter. So basically those disks are very small. And so
so your y-axis is scale parameter and your x-axis is time?
Yes, exactly. And so these colors represent how many connected components, as I said, and that's kind of it's kind of difficult to it's kind of opposite of at least what I think when I hear that word. Really what it is how many things are all globbed together into a single blob. So if you have a bunch of things with the disks overlapping, all these points with disks overlapping, they all kind of turn into one blob essentially. And so as the scale parameter increases, you can see that we're having more things starting to turn into starting to connect into a network or a blob essentially. And so as we get to a certain point, and you can see the connected components here, yellow is a large amount of them, blue is low. So what this means is that everything is becoming connected up here at some point. And so you can see that this starts to happen sooner as time goes on. So this is from a simulation where the cells were moving together so that they would be physically closer together. And so what that results in is quicker connection, essentially, if that makes any sense. I don't know if that helps you.
As your radius goes outward, you're more quickly reaching a connected space because everything is moving closer together, is kind of the idea I should get from this plot.
Yeah.
That's why this window is nice, because you get the same sense of the window, but you're using less frames, essentially.
Yes, exactly. So I think you've got it pretty much exactly then. So I'll scroll back up, and I think I talked about most everything here except for what our goal was. So like you just said, the window is sort of a way of limiting the amount of time frames we have, but we want to keep the level of accuracy, at least a satisfactory level of accuracy in classification. So that was our objective. How much can we eliminate and still have a satisfactory result in terms of accuracy. And so we basically, here's the basic idea. The red lines would represent possible choices of endpoints for a window. And so everything between those would be what it would be a separate SVM from the full one, but it would only see these time frames. So it wouldn't see anything before this, and it wouldn't see the end. It's pretty easy to see from the end that everything would be clustering in this simulation, for example. And so a lot of the time, we were more focused on intermediate behavior in terms of accuracy, but regardless, we basically set up a program that allows us to slide these red lines around and test every single possible combination of the end and start timeframes. And so that could mean these red lines might be overlapping and you only see a single time frame, or the starting red line might be all the way over here and the end one over here near 100, and you see the full thing again. Every single combination of those two lines that's possible was explored by this program. And so we also needed to look at what our measure of accuracy should be. And so the measures of accuracy that are commonly used in machine learning problems are typically sensitivity and specificity, which sensitivity, this one is the proportion of positive classes, that is, in this case, I think it would be clustering, that are actually positive. So it's the proportion of positive cases that were actually positive. So what that means is that if it were to misclassify 10 as positive, but still have 10 positive that were correct, it would be a sensitivity of about a half, which means half of those were actually positive that it classified correctly.
Connor, can I interrupt once? Is it just sensitivity or specificity, or do you also consider positive predictive rate, positive predictive value, and the negative predictive value, or particularly PPV? That's something.
I haven't looked at that. I should probably look at that. It sounds similar, but I'm not sure if it's the same thing.
It is not the same. Sensitivity is your positive by the true positive, as you are saying, and the positive predictive value is that what is the predicted positive, the true positive by the predicted positive. So it will take the false positive also in the consideration. So it's just that 2 by 2. What is that called? The ratio? Huh? Ratio? No, that 2 by 2, that plot that you have true positive, false positive, and
Just the different.
Oh gosh, I know what you're talking about, but I can't remember.
Yeah. It's
The
just
confusion matrix.
confusion matrix. Yes.
Okay.
Yeah.
All right. Yeah, I've been. We were using those values in order to get these measures of sensitivity and specificity. So it wouldn't be too hard to actually adapt it over to get those. Those positive prediction rate, is that right?
Positive predictive value.
Okay.
Yeah.
Okay.
Yeah, we have, I think Marco is also in that paper. They talked specifically about that PPV value. Then I looked at, they look at it with importance for diagnosis of any kind of prediction you make. So sensitivity is definitely one important issue, but PPV also important. Yeah.
Okay. Well, right now, the only measures of accuracy we're looking at are sensitivity and specificity. And usually those are kind of like dependent on what your objective is. Like here, sensitivity is important in if you're using like a classification algorithm for a cancer screening or something. You want to know if the cancer is malignant or not. Well, you'd probably want the algorithm to be really good at picking up on those cancer screenings that are actually malignant, right? You wouldn't want any to fall through the net. And what that would result in is oftentimes it would give false positives, but at least you would have all the malignant ones classified correctly. But those are things you have to weigh when you're working with machines, and often that causes you to nudge the weights of things, and that can start to affect your accuracy as well. But in order to sort of look at those sensitivity and specificity easily, we created a pseudo color plot. And what you can see here are essentially there are, it's an 100 by 100 grid of squares. Everything above the diagonal here is white because those aren't like choices of time frames that you can have. So what this is the vertical axis here is the choice of starting time frame. So you'd be starting from 1 down here, for example, and then the horizontal is the ending snapshot or time frame. And you can see here, if you choose 1 as your starting, you can go all the way up to 100, for example. If you choose, and in every case, you can go all the way up to 100 from that point, but you can't choose anything to the left of those points because that would go before your choice of starting snapshot. And so each square here has a color assigned to it. And those colors correspond to the level of specificity or sensitivity. And so yellow is really good, and it goes to orange as it gets a little less accurate, then to green, then to light blue, dark blue. And you can see as you see less and less over here, this is only the very beginning of the simulation. You only have a pretty weak measure of specificity and sensitivity down here. But you can see there's some interesting behavior in the intermediate areas here as well. Okay, and in addition,
can you just stay for that on that slide
Yeah,
for a second? I have a question here. Let me just. So just to kind of be clear. So when we are, if I'm thinking about my simulation, right, the Crocker plot that you were showing me, right? You have those 2 window bars that were denoting which window we are using, right?
Yeah.
So points here are the window is very small and is sitting in at the beginning, right?
Yes.
Right.
Yeah, pretty much.
Points here are the window is actually very large.
Yes.
And it starts at the beginning and ends, right? And points here are the window is again small, but now it's sitting at the end.
Yeah.
Right?
So the x-axis is the right side of the window, and the y-axis is the left side of the window.
Yeah.
Exactly.
Okay, it makes sense now. Yeah.
Yeah. If you could label x and y axes next time, that would be very helpful.
Yeah, that would probably.
No.
Yeah, I'll make sure to do that.
But even without that, I'm just trying to. I'm in my head. It's clear here. I'm just trying to understand. You know, there is obviously things on the right are all good, right? Which means that we're basically taking in all the data that we have, practically speaking, right? And making a decision based on that. So it seems like if I take the entire Crocker matrix, I'm good, right? I can predict correctly whether I'm clustering or I'm not. If I'm
Great.
taking just, and that also means if I'm here on the left-hand side in this lower corner, that basically I don't see anything, right? That I can't predict anything because the motion there is still very random, right? Nothing started to cluster, so I can't really predict one way or the other.
Right.
So
Exactly.
top right is really good. It's doing a good job predictively because even though you're taking a very small portion of time frames, the data is very clustered at that point. So it's very
Yeah.
easy for the algorithm. Okay.
Yes.
Right. You kind of
exactly.
just, it's kind of like looking at the last snapshot and saying, is this clustered or not, right? Where you don't care how you got to that last snapshot, right? You're either clustered or not clustered. So you can take a very short window. But I guess I'm more kind of interested in this intermediate band, right? Where in that band there is, the sensitivity is really good, right? Which means that I'm willing to take in a lot of false negatives, right, just to get the correct ones. I'm willing to take in more false negatives, right, just so I could actually make sure that nothing is really getting missed.
Right.
And so I'm trying to understand what this means in terms of where the window is, where this band lies, right? So I start something fairly early, right? And it seems like as long as my start point, if I look at these two graphs, it doesn't actually matter where I end, right? Graph doesn't show any variation with respect to the vertical, with the choice of the vertical, right? It only matters where I start my data. That's what distinguishes between a good window and a bad window.
Well, the vertical axis is the start point here, though. I think you have them labeled backwards here.
Oh, backwards.
Because so as you go vertically, and you reach a certain point along the diagonal here, you can't have an end point that's before your start point. So that's why that's how I think about it, as the vertical is start and the end is.
So I can pick a start point at any point, right? So I can pick a start point at any point. Oh, I see what you're saying. I can pick this is the end point, and this is the start point.
Yeah.
Oh, okay, yeah. So I guess then it doesn't matter, right? Whether I guess the images that I drew are somehow still correct, miraculously, but so it matters only where I end, not where I start.
Right, and I mean that does make some sense when we look at how this. I mean, the simulations are only get different later on as they go through time. So I think that does make some sense.
Yeah, because all of our simulations are kind of monotonic, right? They start disordered and they either go to ordered or they don't go to ordered. Nothing,
Right.
There's no simulation that kind of fakes out, right? Like that starts going ordered and then backs out and goes back large, right? So this is really, I think this is really showing us more, this gives us more knowledge about what the simulations are looking like rather than what the test is looking like, right?
This would be interesting on like some oscillating data or something like that.
Yeah, so I guess that's right. So if we have some data that is, if I have points that start disordered, right, and let's say that everything is just here on the 1D axis, right, space and then time, and so these points are moving about, right, so they start maybe moving together, and then they back out, and then they again move together and back out, right, then we would probably we would lose that kind of predictivity, but in some sense, then you lose the assumption that the past looks like the future or the future looks like the past. So you don't really have, like, if anything goes, that points can do anything going in the future, regardless of what they have done in the past, then you can't really predict anything, right? You
Right.
have to have an assumption that the data that you're ingesting is representative of the future.
Right.
Okay, yeah, I guess I wanted to think through this kind of out loud,
Yeah.
rather than move on. And so I guess I would like to understand also why the specificity and sensitivity are not exactly the same in this way.
It may be that the algorithms tend to push one over the other, and I want to look at the differences. I still want to get the advanced diagnostics running for some of the algorithms that, or the models that we're creating.
Okay.
The problem is if you would have to pick and choose, because you wouldn't want the diagnostics for all 100 of the models that are being produced for each of these time frames, you know.
Okay.
So
when you say diagnostics, what do you mean?
there's ways to look at, for example, like what weights it's throwing around on things, what the support vectors are, those sorts of things. And those can be pretty useful at looking at why things are looking like this, for example.
Okay. Okay.
So I have a naive question again, Connor. Is it known in the field for when you do this analysis, sensitivity comes before specificity, or we are seeing this for this particular data set?
I don't know actually if it's normal for them to do that, but
is this the ratio between sensitivity specificity? Is that a function of the model or the data or
Right.
both or parameters in it? So that would be something to investigate, I think.
Yeah.
okay.
Yeah, but it is nice to see visually. I think that is very
Yeah, these
interesting.
are awesome visualizations.
Yeah, yeah.
Okay. Yeah, I don't know if I understood exactly what you were asking. So
So I was asking is that if anything, sensitivity improvement, it's generic in the models that improvement will come first and followed by the specificity improvement. Is that?
I think what Shantanu is kind of asking is when you're fitting the model, is it trying to fit specificity then sensitivity or vice versa, or is that a metric that it uses at all? And that's
I think I see what you're saying. And again, I'm not sure exactly.
because that was what you kind of mentioned earlier, that was something you could kind of choose. So I'm wondering, you know, if that's
Yeah,
something
you can
parameter.
basically weigh something like a false negative at a higher weight, for example.
You're right.
So this pattern that we're observing, could it be induced entirely by the algorithm that you're using and not necessarily by the data?
Right, and that maybe go back into the argument that Dr. Budicic was just making about how our data is very similar in a lot of ways. It evolves and then it doesn't go back to its previous disordered state, as he said. And so I think you may be right there that may be the case. So that would be something interesting to explore as well. And obviously in real cells, they wouldn't just stay in a clustered state over the rest of a time frame or whatever. They might start to eventually move back out. And so, yeah, it is a question about figuring out how to do that with the simulated cells and the model that we have. But it may be something that is possible to create.
Connor, can you just briefly show us what the clustering targets look like for you? Like what are the shapes of those of the clusters at the endpoints?
Gosh.
Or maybe not show us, but just describe. Like, do you remember is this always just going to one cluster, or is this going to different?
No, this does have the advanced clusters in it. So, so clustering can look like, for a simple cluster, it would just be some sort of, well, it would really be a square, and then all the cells would be randomly placed inside that square. And then the advanced clusters are based on PNG images. Basically it takes the image, and for example, I think one of the images looks sort of like this. And it, for example, this might be one of the target areas, these dark areas here. And it essentially takes that image and it throws points at it until it has enough points that are sitting inside of the dark area. Sort of like throwing darts at a dartboard, and letting them stick to the areas where you would want them to. And so it's still random placement, and it does this for the end points of the clustering, so that when you have it moving into its clustering form at the end of the time frame, it sort of clusters into the shape of that image that you fed to the rejection sampling function. And so this is only one example of the ones that I'm using. I think another one is sort of a blobbish shape. And then what was the other one? It was one that was sort of shaped like this, I think. Sort of two clusters, I think. And then I can't remember the last one, but
and all of these targets are mixed both into the training set and the validation set, right?
just the validation, so the testing.
Okay.
Because
And
one
it's
of our objectives before was to test how the trained model would do on things that it hadn't seen before, right?
right.
So it's being trained only on the simple clusters?
Yes, so something like a square where all the points are randomly placed in it, which would result in roughly a circle-ish cluster. But then we also feed it these into the testing sets and see how it performs on those as well.
So when we're done with the meeting, it would be good to actually add a slide about this into this deck, and then share the slides with all of us. I think this is kind of an important piece of information about like what you're trying to do. So because we talked about this, right? Like our first result when Connor ran this, you can think of this as being basically in the lower right quadrant of these images, right? So this was the entire Crocker plot, and the results were perfect, right? Like we could not break this prediction, right? It was always predicting exactly the correct, right? And so we started playing with ideas of like, well, what if we train it on one set of clusters, right, and feed it another set of clusters, right? What if we give it less data in the training and then try to predict, right? So we're all of this, I think Connor's work since the summer was trying to break the overly great result that we got over the last summer. In some sense, right, it's unrealistic that you create a perfect classification, right, that exists only in theory, right? Or if you're rigging the game, right? So we're trying to see how much variance to add both to the training set and to the algorithm that we're using in order to push this to the limits.
And these are also things that are commonly done in machine learning as well, reducing the amount of variables that you're feeding in, looking at expanding beyond your initial problem set to try and generalize, which can have its own set of problems. But yeah, so those are pretty common places to explore when trying to improve a machine learning algorithm.
So one thing I think what would be interesting, so right now you're doing this only with H0, right, with beta zeros?
No, I think it's doing both Betty 0 and Betty 1.
Okay, yeah, so one thing that would be interesting to see if we can actually make this into kind of a multi-class situation, right? Where we say, okay, I want to not only predict whether this is going to be clustering into a single cluster, but whether how many clusters it is going to break into, right? That will be we can look at. And the second thing is that can we actually predict the shape of the cluster in the sense that we can actually classify if it's going to a torus, if it's going to two toruses, right, or so on. So in some sense, how that end shape that we get in the end, right? How soon can we get the actual Betty numbers correct, right? If I'm giving it less and less data initially. So I guess it stops being a classification problem, and it's more like a regression problem, right? Where we're trying to basically estimate the actual values in the Crocker plot, the last slice of the Crocker plot, right? That's the end state, or at last vertical slice. And so we would try to see how much of the initial slices do I have to feed in order to predict that last slice correctly, right? Obviously, if I give it the slice, then it can just take it, right? But I want to know as soon as possible rather than waiting till the end. So that's, I think we said next week in our one-on-one meeting, we're going to talk about like what to actually now zero in on. So that will be one thing to kind of keep in mind of what we might want to look into.
Okay.
Oh, and one actually, you're gonna share this slide. So one thing to add is these targets. The second thing here on this slide is to make sure that the vertical axis, that the color axis on both left and right are the same. Right. I just noticed now that they are not starting at the same point.
Right. Yeah, they are not. You're right.
Yeah.
Which might show some details in here that we would not otherwise see.
Okay.
Yeah.
Nice work, Connor.
Yeah, thank you.
Yeah.
Yep.
I think this was very helpful.
Okay. Thank you. Right. And then we've been working in GitHub as well. So that's pretty much everything, I think. So I'm going to stop sharing.
Yeah, so the thing that we did over the last week was we figured out a way how to run. So the code that computes these persistent homologies, right, is kind of difficult to code up on your own. And so we were using packages from different people, which depended on basically Connor packaging data into text files, sending to those programs and pulling text files from those programs. And that was kind of a lot of hassle. It was very easy to break. And so we figured out a way how to stitch MATLAB together with these packages, so he can now do everything inside MATLAB rather than writing files on the hard drive. And so this kind of brings us a bit closer to the setup that Devasha has in R. This package is not as powerful as hers, but at this point it's it's much easier to work with Garner. So that was kind of the code that we were trying to figure out over the past week. So if anybody else has a preference of working in MATLAB and wants to try this, now we're set up.
Marco, one question for you. How did you set this webcam with focusing on you and?
Oh, it's an artificial. It's like in Zoom. I know
okay.
If you update your version, one of the backgrounds is a blur rather than just kind of a fake.
After I saw you have that for the first time, I figured it was digital, and I was trying to figure out how to do it, and I couldn't figure it out. So I guess I gotta update it.
Okay. Now I thought that Marco has a very fancy lens on the webcam, and
I thought
no.
Too good. I
Yeah,
like
yeah.
too
Yeah,
good.
fancy. Marco
You
has
can
a
see
fancy
like
mind.
if I move my head, right, that it's kind of like
working very nicely. Actually.
Yeah, no, I liked it a lot better than any of the
Yeah.
I don't like the big distracting backgrounds, but I also think it's more professional to have it blurred out, and so I really liked that. So,
Yeah, I liked it very much. Exactly. I'll try that from now onwards. Yeah. Yeah, very nice.
all right.
Yeah.
Well, thank you very much, Connor and everybody. I will send out slides next Thursday so that everybody can get a chance to look at them before Friday's meeting.
Perfect.
Yeah, and I guess I'm still recording. I'll stop recording right now.